Yes, my name is Jiguang Wang. So my topic today is a little bit different because the topic is about big data and complex systems. So myself have the training with mathematics background, but now I'm in the division of life science, and my lab is working on mostly in cancer genomics. Today I'm going to talking about our recent study on modeling tumor evolution using mathematical models, and then talking a little bit about precision cancer medicine. It doesn't work. This is done. Oh. All right. To start about cancer genomics, I would like to talking a little bit about the status of fighting cancer for human beings. So as we know, so cancer is a deadly disease, and in the year 1970s, you see this graph, the x-axis is the survival rate after 10-year diagnosis of cancer. For different types of cancers, on the first line names all cancer, means whatever cancer the patient get diagnosed. So we treat the patient to see how many patients will still be survive after 10 years. So in the year around 1970s, statistics shows that 25% will still live, means 75% will die in 10 years. However, this number increased dramatically in the past few years. In the year 2010, the number already becomes 50%, so indicating if a patient got diagnosed with cancer, he has more than half chance to survive more than 10 years. However, this situation is different across cancer types. You see here on top, some cancers have survival rate around 100%. However, if you're going down this list, you see the tumor on the bottom, pancreas cancer. It's a kind of the king of all cancers, so this kills patients very quickly, several months. And lung cancer, also brain cancer is basically the focus about today, is around here. So brain cancer, so one of the most aggressive type of brain cancer named GBM, also named glioblastoma. So it's a very aggressive type of brain tumor. It's the most common and aggressive tumors in adults. And this incidence of this tumor actually is not quite high. So every 100,000 people per year will be diagnosed with GBM, around 2 to 3 people. So actually, if you do the math calculation in Hong Kong, there will be 100 to 200 GBM cases per year. Without treatment, the patient will die within 3 months. But with very standard treatment, the patient will die around 12 to 15 months, indicating even with very good treatment, patient will eventually die. So this is standard treatment of GBM. So this method actually developed around more than 10 years ago. So in the New England Journal of Medicine, in 2005, Dr. Roger Stupp developed a new therapy using a drug named temozolomide together with radiation therapy, surgery, and put together to get this blue curve. You see the blue curve has a little bit improvement than the red curve. The patients in the blue curve have around 3 months more average survival. You can see the difference as around 36 months, around 3 years. So the survival of the blue curve is about more than 20%, but the red curve is almost 0, indicating this drug actually works. So the mechanism of how this drug works is because this drug is alkylating agency. This one can alkylate the DNA, can trigger the death of tumor cells, and so the cancer cells can be killed by this drug, and the patient will have better survival. However, even under this treatment, in the blue curve, almost all patients will recur. Eventually, the patient will recur. Even the patient have a longer survival, the patient have another tumor again. So when the tumor recur, there's no standard treatment, not yet, even for today. So to study the mechanism of GBM, so large sequencing projects initiated by NIH in United States. So specifically, the Cancer Genome Atlas project actually sequenced a lot of number of patients. In particular, in this study, they sequenced around 500 primary GBM patients, and they found the genomic mutations in cancer cells. But those mutations are not in the normal cells. indicating if you go into details of the cancer cells, you find out that in the cancer cells, you see the mutations in all those genes here, but in the normal cells from the same patient, there is no such mutation, indicating those mutations somehow can serve as the marker to identify the difference between cancer cells and normal cells. Therefore, we can develop some treatment to particularly target the cells who are carrying those mutations, indicating those landscape study can provide us a lot of drug targets. So here, I'm only mentioning several genes, EGFR, MET, FGFR. All those genes have particular small molecular which can target the protein domain and eventually kill the cells. However, in the trial of those GBM patients, so almost all the trial failed so far. So our work mostly focuses on the cancer evolution process. We want to explain why the trials will fail and why the patient will still die after very intensive treatment. Our major hypothesis is that the cancer process is actually a evolution process. So the cancer cells was starting from a normal cell, and after the growth and accumulation of mutation, the cells will develop different clones. For example, there's clone number 1, clone number 2, and at diagnosis, we can cut the tumor, and we can do the sequencing. We can identify the mutation in the cancer cells. Also, we can do the treatment on the cancer patients. We can do the radiotherapy, we can do the chemotherapy, and so on. As I mentioned, for GBM patients will recur. Eventually, they are going to die. So our question is, if we take another cancer sample from the recurrent tumor, whether the recurrent tumor should be identical to the primary tumor or not. So if they are identical, then we can use the same method to targeting the same genes at the recurrence, right? If they are different, then our method mentioned before will fail. Okay? Based on this logic, so we designed several studies. Study number 1, we did a longitudinal study. From different time points, we sequenced different samples, and then we compare the difference. Eventually, we found out from 100 patients, we found the new mutations at the recurrence. For example, the mutations in MGMT, LTBP4, and so on. Also, we found out the mutations have an order. The order means if you find one mutation at the first time point, usually you have a more chance to find another mutation at second time point, and that mutation is kind of predictable. In the second study, we studied the spatial information of the cancer cells. We can take the cancer cell from the same patient at the same time point, but different location. Also, we can monitor the difference between different locations in terms of genomic mutation. So in the third study, so we studied the gene drug map, indicating if we have a cancer cell and we treat the cell with different drugs, we have the outcome for this cell and this drug, right? So we have a lot of features from the drugs. We have a lot of features from the cancer cells. Then we can combine the features to predict the outcome using mathematical model. So this is our third study, so published in this year. So, but today I'm going to talking about a different story, basically the approach in my laboratory. So then my PhD is from mathematics, as I mentioned, and now I'm doing a precision medicine and computational genomics study. So our goal is to cure the cancer using formulas or using computation, using high throughput computation. And today I'm going to show one example, how can we use the technology of big data, how to combine the research from hospitals, from a research center, and from data scientists together to fighting brain tumor. To start, I'd like to introduce another rare type of GBM. So GBM, as I mentioned, glioblastoma. So actually, if you go into details of all glioblastoma patients, there are majorly two subtypes. One is named as primary GBM, takes around 90% of all GBMs. Another subtype is rare. It's named as SGBM. This SGBM actually has the history of a low-grade glioma, indicating that patient was initially diagnosed with a low-grade glioma, usually grade 2 or grade 3. And then after around 10 years, so the patient will eventually develop into grade 4 GBM. So for grade 4, the survival is very low. It's around 1 year maximal, not maximal. So on average, it's 12 months So, but this is a very aggressive tumor, but this is less aggressive, indicating this patient has around 10 years survival. However, so because this type of tumor is super rare, as I mentioned, GBM is already rare. This is only 10% of GBM. So very few studies has been focusing on this IGBM, and very few Asian gliomas has been carefully studied. Because now we are in Asia, in China, we have a huge population base. So based on the population base, we can actually get a lot of cancer samples from this rare type of GBM. So in this case, I would like to review several studies. I already published some papers on the SGBM cohort. The first study actually from China, from Beijing Cancer Hospital, Jiang Tao's group. In this group, in the year 2014, so they published a paper about 20 SGBM patients. In those patients, they identify new gene fusion, indicating these two genes should be isolated normally. However, in those patients, those two genes go together, and one of the MET gene is a very famous oncogene, and this fusion will make the oncogene super active and leading to the progression from the low-grade glioma to the high-grade glioma. Because I mentioned low-grade glioma is a glioma at a grade 2 or grade 3, but the GBM is grade 4. So this is very aggressive, and to understand the mechanism, so this study indicates that this fusion is very important. In the same year, published in Science by a group from UCSF, and they identified a different mechanism. They studied 10 SGBM patients, and finally they found out a phenomenon named hypermutation. Actually, it's leading the patients from LGG to SGBM. In this study, so in particular, you can see clearly here that there are 6 patients. So each block is 1 patient, and each column is 1 sample at 1 time point. So at the first time point, if you count how many mutations in this patient's cancer cells, the number is around 10 or something. And in the second time point, still a very small number. However, at the third time point, the patient developed into grade 4 GBM. So this patient carrying 2,500 mutations. So similar story in different patients. So this 6 patients in total. So you can observe similar things. So like at third time point or at the grade 4 GBM. So those patients have more than 1,000 mutations. So this phenomenon named as hypermutation. So because of this phenomena, so they think the mechanism from LGG to SGBM is because of the hypermutation phenomena. So it's different from the MET hypothesis. The third one came from the year 2016. So again, they have different hypotheses. They think MYC pathways and the RTK, RAS-PS3 kinase pathway is very important from the study of these 17 patients. So another study from our group, my previous group in Columbia University, actually, when I was doing postdoc, we are publishing this paper in Nature Genetics in 2016. So we found out actually 1 gene fusion in the SGBM patient. This gene fusion involved the gene in MGMT. So at that time point, we think it's because of the fusion of MGMT. So far, you heard about 4 different stories. So this is a famous cartoon shows that there actually every study has different view because they are studying only around 10 or 20 patients. So they find totally different mechanism leading to the evolution of GBM from low-grade glioma to high-grade glioma. So then how can we know the whole picture? So our method is big data collection. So when I came back to China, I talked to the largest hospital in Beijing, so the Beijing Cancer Hospital. So actually they collect the largest cohort from the Chinese population, and they do surgery, more than 1,000 surgery of GBM every year. So in their cohort, they can easily get the number 108. Even we collect 10 papers, 10 different papers. So all those papers are high-profile papers. Nature Genetics, genome research, cancer cell, science, cell, and so on. All those papers contain only less than 20 patients. However, from Cancer Hospital, we can easily get 108. So with all those patients, we got 188 patients, and we got the sequencing data from all those patients. Then we can have a global picture of the disease, SGBM. So this is our result. So we found out that in the SGBM, actually all the stories are somehow correct, but there is a global picture. You can see the mutation in IDH1, P53, ATRX, and so on. I'm not going to mention too many gene names because the audience I know is like more from like a mathematical background. But I would like to mention one observation is the difference between Asian and Caucasians. So actually, the hypermutation, as I mentioned, is more common in Caucasians, but less common in Asians. So the reason for that, so there are many hypotheses. So it could be the treatment, could be the like difference, like a disparity, their like a racial disparities. It could be many others. However, we do observe the difference, so indicating maybe for the treatment, we should also use a different treatment method for the Asian patients. So also we identify some mutations, like a new mutation, for example, the mutation in MET exon 14 skipping. So this mutation actually happens in 14% of those cohorts, and it turned out it's one of the most enriched mutation in the SGBM cohort. So when we compare different cohorts like LGG, like a primary GBM and secondary GBM, we found out that if the mutation in MET is very enriched in the SGBM cohort. So this triangle is basically the percentage of the mutation in which subtype. So I mentioned there are three subtypes. LGG is a low-grade glioma. SGBM is the GBM developed from low-grade glioma. And primary GBM is the GBM. It is grade 4 as diagnosis. Okay, because this is three different types of disease, we can do the enrichment analysis to see which genes are enriched in SGBM. So the result turned out the MET exon 14 mutation is the most enriched mutation in SGBM, happens in around 14% of patients. However, the same mutation happens only in 1 to 2% in the low-grade and primary GBM. To be more clear about this mutation is actually a deletion of one exon in the MET oncogene. So then we performed a series study about this MET protein mutation. We found out the deletion of exon 14 actually, like the consequence of this deletion is the removal of the GM domain in the MET protein. This is a GM domain, and in the deleted version, the GM domain lost. Okay. So this study already been reported in the paper published in 2016 in lung cancer. So now we identify the same mutation in brain tumor. So then our collaborator from Beijing Tian Hospital performed a series of a functional study. So we found out that if the patient has this mutation, the patient will have high expression in MET, also have the activation of the downstream pathway, for example, the STAT3 pathway. Also, we verified our hypothesis in the cell line level, also in the patient sample level. So more importantly, we found out that this mutation can actually mark the worst survival of SGBM patients. If you read this curve, you see here there are two curves, one in blue, one in red. The blue patients indicating the patients with the MET exon 14 wild type. The red patients indicating the patients have the mutation in MET exon 14, indicating this mutation actually can drive the progression of the disease and eventually make the patient have worse survival. So then we check whether this mutation happens in the DNA level or in the RNA level. So we find out that it's in the DNA level. This mutation can actually, it was because of the change of the DNA from the patient cancer cells. So lastly, I would like to address the question, how can we use this research to help patients? Because this is, anyway, it's our eventual goal. So we do want to translate our research into the clinic to help patients. To do that, we have to review the process from the low-grade glioma to high-grade glioma. So let's assume this is one patient with diagnosis with low-grade glioma. So you can clearly see from the MRI, we can see the tumor box is over here. So usually the patient goes to the hospital. The first choice of the doctor will be do the surgery. So do the surgery. Because of the surgery, we can take the tumor tissue to see what happens in the cancer cells. We can do the sequencing. We can do the pathological evaluation and so on. Many things we can do here. And after the surgery, the patient go back to do the MRI. You can see clearly the cancer cells is removed. There is a blank hole over here. So, however, unfortunately, for most patients, after a while, the disease will come back. So a couple of years, typically for low-grade glioma. So when the tumor recurs, you can see again in the same location. Sometimes it's even in different location. You can see the recurrent tumor over here. So then that will be a big question for the patients, for the doctors, for researchers, everybody. What should we do here? So many possibilities. It could be do another surgery, but it depends on their family status, depends on the view of this patient, because even after another surgery, the patient might be able to survive for another couple of months, but it will have a lot of danger in infection or also the surgical cost and so on, many issues here. So then another choice is do the TMZ. It's a standard treatment. Radiotherapy, chemotherapy plus the TMZ is a standard treatment. But also, some patients will give up eventually. So because at this status, a recurrent tumor is very difficult to treat. Now we don't know how to treat the recurrent tumor. So also, our idea is, do we have other approach or not? So if we can actually know the mutation status at this time point, can we use the mutation over here to help the patients to design the targeted therapy to target the mutation over here? This is our question. So to answer this question, we have to understand one thing. So the cancer actually evolves. So as I mentioned in my introduction, so if you check tumor samples from 2 time points, so time point number 1, time point number 2, the color of the cancer cells might change. The color indicating the mutation. For example, at this time point, the color is mostly red, but maybe in the future it becomes blue. Okay? If we design the treatment based on the red color, it will fail for the blue cancer cells. So in this sense, we have to consider the evolution process, and we have to determine the rule of the cancer evolution. For example, if we observe the evolution of the cancer cells in one patient, and in second patient, and in many patients, actually we can learn a rule of the cancer evolution from those cohorts. And based on the rule, we can design a smarter treatment for those patients. And then we actually did the data collection from a panglioma status. We do the collection from the literature, from the public, the free data. Also, we do the collection from our collaborator from Beijing Tian Hospital. Eventually, we got 265 pairs. Pairs means we have the cancer samples from the initial tumor, also from the recurrent tumor. Then we can compare the difference. We can predict the trend. And based on this cohort, we are able to predict the order of genomic mutations in the glioma cancer cells. In different subtypes of gliomas, we can see the order of mutations. Here we find out that the meta alteration we identified always happens at the end of the tumor, indicating this is kind of late mutations. So then we go into details about the patients with meta exon 40 skipping. We trace back to the initial tumor. And this is SGBM, and this is primary low-grade glioma. And we found out that mutation is actually observable at the first time point. Observable means we can identify it, but the allele frequency or the frequency of that mutation is quite low. So you can see from this curve, it's always increased. So indicating if the cancer patients carrying this mutation at the initial time point, and this mutation is actually exon 14 skipping, then we can predict this mutation will become dominant at the recurrence. So with this logic, we can check also the survival of the patients. It doesn't matter if the patient have a clonal mutation or subclonal mutation in MET, so the patient will invariably have a worse survival. With this idea, we think that this MET exon 14 is actually this blue mutation. So with very small clone, but during the treatment at the recurrence, the blue cell will become dominant eventually. So with this logic, if we got a sample from the first surgery, we find out this mutation, then we can predict when the patients come back to the hospital, we can actually target meta alteration at the recurrence. So this is our logic. And with this logic, so Beijing Children's Hospital carried out a clinical trial to targeting like a MET alteration in SGBM patients. So in this trial, we enrolled 18 patients. These 18 patients taking different doses of the drug. This is the standard 3+3 trial with different doses here. And in those patients, we observed 2 patients with partial response. I'm sharing you with the data about like these 2 patients. Patient number 1, this patient actually with very long history of fighting GBM. So as a diagnosis, this patient is like a low-grade glioma, and the first surgery happened here. So we find out there's mutations in the METX14 in these patients. And after the standard treatment, because before the trial, we have to try many times with standard treatment. After standard treatment, so the patients have some response at the beginning. However, at this time point, the patient stopped to respond. So the response just means like 2 things. One is based on MRI, do we see the shrinkage of the cancer cell or not? The second thing is like whether we see the relief of the symptom or not. Okay? If there's no response to this drug, the standard treatment, we have to stop the treatment, and then the patient choose to enroll in this trial using this new drug, also identified by Beijing Children's Hospital, with 5 cycles. And in this trial, taking around 20 weeks, you can see the tumor from the baseline is this one. So you see the recurrent tumor over here. At the 4th week after taking this drug, the tumor disappeared. 8 weeks, still not there. So also at the whole period, the patient has the symptom relief. So however, at 20th month, the tumor came back. And then this is the end of this trial. The patient eventually died at this time point. So then, I show here, there's another surgery over here. So we actually take another tumor sample from the same patient, but after the trial. So then we have tumor sample over here and over here. Then we can check the difference between before and after the clinical trial. So interestingly, we find out another new mutation actually happened in these patients, the mutation in PIK3CA. And also with the RNA-seq, we find out the activation of PI3K pathway, indicating although the patients have the response to the trial at the beginning, so a new mutation makes the cancer cell become resistant to their previous treatment method. To summarize, so we characterize the mutation landscape of secondary glioblastoma as GBM. It's a very bad brain tumor. And also we find out MET alterations can recruit the macrophages and promote glioma progression and mark worse survival. Also, like our collaborator identified the PLB101 is a highly selective, efficient, and blood-brain barrier penetrable MET kinase inhibitor. Eventually, we proved that this drug is a safe and useful treatment approach for glioma treatment. Finally, I would like to thank the people in my lab who are doing the computing work in this project. Also, our collaborator from Beijing Children's Hospital, collaborator from Samsung Medical Center, and also thanks to the advisors' helpful discussion from Columbia University, Robert and Antonio Yarani. Thank you.
Small number of genes in general showing up in these later cancers, is combination therapy potentially an option? So just try to hit at the very beginning all of the things that are at least a subset of the things that are likely to come up.
Yeah, that would be the ideal method, but it's have some ethical issue. Because according to the protocol of the doctor, they have to use the standard treatment. So because you cannot make the trial at that time point.
So is there, I guess this is showing up in a number of different diseases like antibiotic resistance and microbes, the problem that one needs combination therapy for evolution reasons, but it's not part of the protocols. I'm curious, the United States is completely jammed. I mean, they're not dealing with it. I'm curious if in the Asian countries, this is, it looks like this might be more possible to address.
No, so according to the doctor, they're following their protocol, so you cannot do that, right? Yeah.
Regarding the number of data points, you show that you're able to analyze far more data points than the number of patients in literature, but still it goes from what, 18 to 200. What is in the way of looking at thousands or more? What prevents you from collecting that information?
So firstly, it's very expensive. And secondly, so now there's a big issue for the data sharing for the go outside the country. So in different countries actually, so Korea, China, now we are trying to do the collaboration, but trying to de-ID the patient. So because based on the mutations, yeah, for the patient, you can easily ID that patient, right? But how to de-ID it is a big problem for the genomic field.
Thank you.
Since 19 have different mutations, which kind of mutations you use?
Sorry, can
for the mutations you use for the cancer evolution, because there are many different kind of mutations. Do you treat different mutations at the same time for the future? And also, since you know that exon 14 maybe is some feature that distinguishes the cancer from different stage, but how do you know this gene is to be used for treatment?
Right. So that gene actually has been very well studied. I didn't show the data here, but we did the study in the cell line level, in the mouse level, and in a different patient-derived cell line level, and many studies showing that treatment can actually against the cancer cells carrying the same mutation.
For the exon 14, because you use 8 patients and I use some treatment to target that exon, that gene, but only 2 patients respond. Eventually patients dies. That indicates exon 14 is not the one to be for target trial.
No, it's very complicated. So here, so for the clinical trial, so if you see more clearly here, so here there are 3 types. One is partial response. It's not totally response. It's only partial response. Then a second type is a stable disease. Third type is a progressive disease, indicating 7 patients after taking the drug become stable. So if they don't take the drug, they will be progress quickly. So basically 7 is stable, and 2 have the partial response, and 6 is still the tumor is not stopped. So the model can only explain partial of this story. So for those disease, in the patient, it's super complicated. It depends on their genomic background, depends on the purity of the cancer cells, also depends on the real status of that patient. Also, you see here, there's 3 patients. They didn't continue their experiments because they withdraw in the middle for other reasons.
Okay, so stop here and then you can answer this part.